Good morning. My name is Doug Melton, and I'm very pleased to be here to greet you all for your 25th, 35th reunions, etc. I'm grateful to you coming in this morning on such a beautiful autumn day and sitting inside. We're going to be together for a little more than an hour, and I hope you won't think of this like your worst high school biology lecture. But instead, I'm going to talk to you about my favorite subject, stem cells, and I'll show you about 30-some slides by way of introduction for our discussion. The main point of my talk today is going to be to try to convince you that there's enormous promise in stem cell science, and there's been quite a lot of progress in the last few years. At the same time, there are significant political and legislative challenges to doing this sort of work, and at the end of my talk, I'll directly address those issues. But I'm going to begin by putting stem cell biology in context for you, then tell you why scientists are so excited about stem cells, and talk to you about what we've been doing here at the Harvard Stem Cell Institute, and then look a bit to the future about what I think we could all hope to hear when I see you for your next reunion. So I'm going to begin by reminding you about how the public in general thinks about biology in recent days. At the beginning of the last century, there were important discoveries by Mendel about genes, something you all know about. And then throughout that century, it was possible to determine that the stuff, the chemical of genes, was DNA, and practically every kindergartner knows that DNA is a double helix. At the end of the last century, the entire sequence of the human genome was determined, and the public has wrongly, I think, concluded, largely because of the way newspaper articles are written, wrongly concluded that what makes us who we are is our DNA. And I'll remind you that DNA is important, but it is not the be-all and end-all of who we are. In fact, our interactions with the environment determine what we become and how we survive. And I'm going to make the argument to you today that DNA is not really the most important unit of life, but it's actually a cell. And of the cells, the sort of mother of all cells is a stem cell. And I'm going to try and convince you that as we look forward to this coming century, the stem cell will become the focus of most of all the biology and medicine, and it will lead to new drugs, new ways of being human, and new ways of repairing our bodies. So for my first sort of conclusion, I'm going to make an argument to you then that the 21st century will focus entirely on stem cell biology and regenerative medicine. Of course, technically speaking, it's not possible for all biologists to do this, but one will see a greater and greater emphasis on stem cell biology in the years ahead. Chris, I wonder if I could ask you, I hear some humming, and I don't know if it's, yeah, that's great, thank you. I also don't need to tell you all that while the biology of this might be interesting, and as I'll explain in a few minutes, it's a complicated subject because it touches on a number of areas of modern life. It raises questions about when life begins, which leads us to a complicated political arena and public policy issues that I will speak about. There are legal issues related to that, and of course there are enormous business opportunities if we're ever going to make this science come to people. That is, it has to be commercialized if it's going to have any effect on us. So I'm going to start with the context in the middle here of biology. What are stem cells and why are scientists so excited about them? And that falls within the general subject of what's called developmental biology. How does an animal, or in this case a human, develop from an egg? And you'll remember from the biology classes you did take that there are a number of things, most of them obvious, that are involved in that. The first is the egg has to divide to make more cells, and the second is those cells have to become different from one another. You have to have bone cells, blood cells, muscle cells. That process is called differentiation, as cells become different from one another during development. And the subject of stem cells speaks both to this question of differentiation and renewal. So in this picture, you see a cartoon of a human that I'll use a few times in my talk. But the point of this picture then is to remind you that as a fertilized egg begins its division to many kinds of cells, these cells are specified or determined to become different kinds of cells in the adult organism. So here you see some blood cells, some nerve cells. This is the tissue I'm most interested in, the pancreas, which I'll describe a little bit later. to give rise to all the different body parts. But of course, making the organism isn't all that has to be done. The organism has to be maintained. And that maintenance, that turnover, that repair is accomplished by stem cells. So I'm going to tell you about the two main properties of stem cells. One is their capacity for renewal. Stem cells are unlike most cells in your body in that they can make exact copies of themselves with no change in their state. So you could think of that like Xeroxing or cloning. It makes an exact copy of itself, and we call that property self-renewal. It's sort of obvious then that if we could understand the genetic program for how a cell renews itself, that's going to deeply inform our thinking about how tissues and organs are renewed. How do we maintain our skin in good shape? How do we repair our muscle? It has to do with this renewal of a body that's already been made. The second property of stem cells then, so the first is self-renewal. The second property is that stem cells can differentiate or specialize, as I said earlier, into a number of types of progeny or daughter cells, as we call them. And a stem cell that most of you will know about is sometimes called a bone marrow stem cell or a blood stem cell. And that's depicted here, where that stem cell can give rise to all the different components of the blood. The reason I think you might know about it is you will have heard of patients who've had a bone marrow transplant. They're suffering from a cancer. Bone marrow is removed and they're treated with chemotherapy or radiation therapy. And then the blood stem cell, the hematopoietic stem cell, is returned to their body after the therapy, and it replenishes or repopulates all their blood. So here you see all the different kinds of blood cells, the most common one being here the red blood cell that binds oxygen, which is why you're breathing, and these are various kinds of immune cells. Now, that's true for the blood stem cell, namely that it can self-renew and differentiate. But blood stem cells can only make blood, and that raises the question of what do we do about other tissues in our body? And that's where scientists became most excited in the last decade or so, because we discovered a special cell called an embryonic stem cell, which can renew like all other stem cells, but most importantly, it can make any part of the body. So that sounds like a fantastic statement to make. How could one cell make any part of the body? And I can tell you that in mice, that we can demonstrate unequivocally that that cell has the capacity to make an entire mouse. It can make any part of the mouse. And a few years ago, the human equivalent of that cell was discovered. So embryonic stem cells then can renew, but they can differentiate into any part of the body. They can act like a blood stem cell. They can make all the parts of the nervous system, including the brain, and they can make my favorite tissue, the pancreatic islet, which secretes insulin. So the first conclusion I want you to remember is that embryonic stem cells are truly special. Only embryonic stem cells can make all kinds of cells. Adult stem cells, when they exist, are restricted. I gave you the example of blood stem cells. Blood stem cells make blood, but they don't make brain or skin or muscle. They only make blood. And finally, something which is important, not much discussed in newspapers, is that not all adult tissues have a stem cell. So for example, this one I study most, the pancreas, does not. In fact, I don't believe there's an adult stem cell for the kidney or for the lung or for many tissues. And so if you want to think about repairing adult tissues, and there is no adult stem cell, at the moment, our best hope of doing that is using these embryonic stem cells. So I'm going to now move to sort of beyond the biology of stem cells to what scientists want to do with them, and I'm going to talk to you about three things. I'm going to talk to you about using stem cells to replace body parts, using them as tools or reagents for drug screening, and then I'm going to follow that with talking about the future and healthy aging. So in terms of replacing body parts, what would be the body parts one would be most interested in? Well, if I asked you to vote, you'd all probably have a different favorite part of your body you'd like to replace. But let's look at the sort of dark side of it for a moment and think about degenerative diseases. I'm going to put these all together in a bucket and just call them degenerative diseases. But they're the diseases from which I would say our society, particularly our aging society, is now suffering from the most. And these diseases have a few things in common. Number one, none of these diseases are caused by a single genetic defect. It's not as if you can inherit a gene which will then cause you to get that disease. The diseases are all multigenic, meaning that it's the combination of genes you inherited from your parents that incline you to get the disease or not. At the same time, even if your genes kind of point you or incline you, predispose you to a disease, there's an environmental signal. In none of these cases is it known, but there's an environmental signal which pushes you over the edge to get the disease. Now, the reason we know that is there are many cases of identical twins, meaning they have the same genes, where one has the disease and the other doesn't. The third thing that's in common about all these diseases is, to a first approximation, they all come crashing down on a single cell type. That is, one particular kind of cell in the body becomes dysfunctional or destroyed. Alzheimer's, it's the forebrain basal neurons. Parkinson's, it's a midbrain neuron that makes a chemical called dopamine. In Lou Gehrig's disease, amyotrophic lateral sclerosis, it's the motor neurons, the neurons that attach to your muscle and make you move. Cardiovascular disease, it's the heart. In the type of diabetes I study, it's the pancreatic beta cell that makes insulin. But I'm putting these all together to say that what they have in common is complicated genetics, an unknown environmental signal, and they crash down on one kind of cell. But remember, I just told you that there's a very special cell, the human embryonic stem cell, that can make any part of the body. So a high school student would connect those dots. They would say, look, if you have diseases where cells are missing and you have a cell that can make any cell in the body, why not explore its use to repair patients that are suffering from those diseases? I'm going to give you two examples of this, the one I work on, which is diabetes. So here's a picture of human islets. The cells that make insulin are stained red in this picture so that they can be visualized. And so this little sphere here contains about 1,000 of those cells. These are cells that respond to glucose. If you had some donut this morning, there's sugar in your blood. Your pancreas reads the amount of sugar in the blood and then secretes insulin so that your body can make use of that food source. These islets here are ready for transplantation into a type 1 diabetic, a diabetic that doesn't have any of these cells anymore. And these cells have been isolated from 2 cadavers, 2 dead people that have donated their organs for transplantation. At the present day, it's possible to treat someone and nearly cure them of diabetes by transplanting human islets from 2 cadavers. Now that obviously is not going to work in terms of the economics or even the mathematics of finding enough cadavers soon enough to transplant into patients. So our goal is to make those cells. We want to make enough of those cells to transplant them into people with diabetes so that they'll now control their blood sugars properly and they won't have any requirement for the daily blood glucose checks, the finger pricks, nor the insulin injections that they need to stay alive. And the way we're doing that, I'm going to show you in a kind of simple cartoon form. My lab makes fun of me for the way I show this, but it's actually how I think about the problem. We begin with a cell that can make any part of the body, and now we want to educate it to become a pancreatic beta cell. And so the analogy you might think about is sort of a kindergartner could become anything, could become an NBA basketball player perhaps, or a Harvard Business School graduate. Part of what they become depends on their genes, but an enormous part of it depends on their environment. So we affect the environment of these cells by giving them signals telling them what to do. So the lab, as I said, teases me and calls this my Skittle diagram. Here we're beginning with an embryonic stem cell, and a cell that's almost its equivalent that I'll describe in a few minutes, called an iPS cell. We have to first tell that cell to become part of the gut tube, which is called endoderm. That's the tube that made the innards of your body, essentially what you remove when you're filleting a fish. It runs from your mouth to your anus. It takes all of your lung, your liver, your pancreas, your stomach. All of that comes from the endoderm. We then have to tell those cells to become pancreatic cells, And then within the pancreas, we have to tell them not to make digestive enzymes, but instead to make the hormone-producing cells, the endocrine part, and finally to make a beta cell. Now, I could show you pictures of these cells. In fact, they are colored because the way we do these experiments is we genetically manipulate the cell so that it turns a different fluorescent color when it's moved down the right path. In other words, it becomes yellow if it's endoderm or blue if it's mesoderm. So then we take the yellow cells and start over. And this process, which is a systematic approach to the problem, is going to take years to complete. But I can tell you that after working on it for some 5 or 6 years, I think we're about halfway done. So I now see the end of the tunnel where we will be able to make beta cells. I can't say for certain when we'll do that, but there's nothing we've learned that tells me we will not be able to make beta cells. I mean, better said, we will make beta cells in the next few years. But since these cells don't do much in a dish, I'm going to show you a different kind of cell that we can make in a dish, and that's a heart cell. So I like this experiment because it's the one that we have the undergraduates do as well, because it poses a puzzle and also shows the promise of stem cell science at the same time. So in this cartoon up here, we have a petri dish with little colonies or mounds of cells. So that, for those of you who didn't do science, that would be like when you have mold on your bread because you've kept it too long and it forms in spots. That's because there are many copies of cells that are growing in one place. These human embryonic stem cells will grow and self-renew in a petri dish. In fact, it's interesting to note that we can get them to renew forever. They are truly immortal. They do not change their properties. So we have mouse embryonic stem cells that have been growing for up to 3 decades, so 30 years, and a mouse you probably know only normally lives about 2 or 3 years. So the cells are truly immortal. And that arrow on the top means that we know what culture conditions to grow them in so that they self-renew. But if we take those culture conditions away, the cells spontaneously specialize, they differentiate. So this is the puzzle for scientists, is how do they know whether to become red, yellow, blue, or purple? How do they know what to do? But we can encourage them to do certain things, and I'm showing you here in the bottom, cells becoming human heart cells. So that's a pulsating heart cell, thousands of them, actually millions here, and these are 4 different examples of them. This, in a sense, is really quite a spectacular result. It's something I wouldn't have dreamt possible when I was an undergraduate. So what you're looking at is outside of the human body, a human cell making a human heart cell. Think about the significance of that. That means if you want to study anything to do with the heart, you don't have to do it in people anymore. And you don't have to do it in a rodent, which may not be like a person. So you're studying the cells you're most interested in, and you're doing it in a petri dish, which means you can manipulate it any way you want. You can do it over and over again. So as I say to the undergraduates, their challenge is to think about that arrow. How is it that you tell a cell what to do? And then how does it maintain that new fate, that new state once it's become a heart cell? And I'll show you in a moment that we can make the starting cell from patients that have different kinds of heart diseases, and then we can study in a dish what makes them have that disease, what goes wrong with those cells. So let's go back to this slide about if we can make any kind of cell, how would we do it? Well, we want to make forebrain neurons to study Alzheimer's, midbrain neurons, the dopamine neurons for Parkinson's, motor neurons for ALS, which I'll show you in a minute, and I mentioned type 1 diabetes and beta cells, and I just showed you the heart cells. So that would be the long-term vision of regenerative medicine, that you would make cells in a dish and then put them back in the patient to repair any cells that were lost by disease or injury. That, I think, is years away. In fact, I'd guess it's close to a decade away at best. But what's a much shorter-term win here in this field is using stem cells not as a product, not to put back in the patient, but to use the cells as tools or reagents to discover drugs. And I'm going to give you one example of that. That's the thing I think we've invested most of our resources in at the moment and where we've made the greatest progress. So I'm going to talk about 2 diseases where the nerves that move your muscles, that make all of your movement possible, die. This first disease, amyotrophic lateral sclerosis, or ALS, was made famous because Lou Gehrig, the baseball player, suffered and died from that disease. And there's a childhood version of it that's very similar, abbreviated SMA for spinal muscular atrophy. At the Harvard Stem Cell Institute, we've been very interested in trying to use stem cells to find ways to slow the progression and eventually stop the degeneration in those diseases. And the way we're going about it depends on a new scientific advance that I'm going to tell you about in 2 slides. But I'll tell you ahead of time what it is. We've discovered a way, as a community, the community of scientists have discovered a way to take adult cells, skin cells from patients, and turn back their clock into this embryonic-like stem cell. It's not perfect yet, but we think that that approach is going to be very useful. It's certainly already very useful for drug screening, and eventually it may even eliminate the requirement for human embryonic stem cells. But the progress in the field has been enormously fast in just a few years. So it started with the work of Shinya Yamanaka in Japan, where he showed that you could take 4 viruses and infect a skin cell, which is called a fibroblast. This is the kind of cell you would have a doctor remove if you go in for a skin biopsy, and turn it into a stem cell. The S is for stem, and this is called induced because the viruses induce it, and it's pluripotent because it may not be totipotent, which means can do everything the way an embryonic stem cell can. This was a very important finding just recently in 2006, and it had created a lot of excitement because we could make these iPS cells, which were much like embryonic stem cells. The problem here is twofold. One is, by creating the cell, one had to use viruses. The viruses there's their main component, DNA, which inserts itself into the DNA of the fibroblast, which is a long way of saying that it genetically modifies this cell. So this cell is no longer exactly like the patient's cell. And secondly, of these genes here, 2 of them are known to cause cancer. They're so-called oncogenes, cancer-causing genes. So in terms of putting these cells back in patients, no one was able to even contemplate that because it involved cancer-causing genes and genetic modification. Nevertheless, it sort of like that joke about if it looks like a duck or smells like a duck or something. These cells were close enough to being embryonic stem cells that people were very excited to pursue this approach. And just this week, you may have seen in the newspaper a major finding here from the Harvard Stem Cell Institute by Derek Rossi, where he's found a way around these 2 problems, and that I just summarize here. The first is he figured out a way to use RNA instead of DNA, and the advantage of that is the RNA, once it goes in the cell, is degraded, and there's no genetic modification. This means that he could make iPS cells with no genetic modification, and there was a bonus that couldn't have been expected, that it greatly improved the efficiency. I didn't tell you that to make a stem cell from your skin cell is a very inefficient and time-consuming process, and Rossi's method improves that by about 100-fold. So if we go back now to this puzzle of how to make stem cells from patients, we now use this method, and we can make stem cells from, if you like, every person in this room. So you could all have your own stem cell, which is capable then of making any part of your body. Now, what do we want to do with that? Well, I mentioned before that for some people, one might want to transplant specialized cells back into the body, but what we're most excited about now is using the cells as tools. That is, tools to get at the root cause of these very complicated degenerative diseases. And that's what I'll show you here in this one slide. So imagine now that we have an embryonic stem cell or an induced pluripotent stem cell from a patient that has ALS. So a patient comes into the doctor's office, they have Lou Gehrig's disease, one does a skin biopsy, and within about 2 months, we can create a stem cell from that patient. This then is our control, or normal, or what scientists like to call the wild type version of the cells. These cells are then differentiated in a dish. You'll remember I showed you heart cells and I talked about pancreatic beta cells. We differentiate them in a petri dish into motor neurons, the cells which are defective in ALS. With Lee Rubin's help now at the Harvard Stem Cell Institute, we've set up a very big program in chemical screening or therapeutic screening So we put the cells in these like bingo cards. They have 384 wells in each of these plates. We put all of the same cells in, and now we add a different chemical to each one, and we ask the simple question, can we find a drug, a chemical, that slows down or prevents the degeneration of that neuron? And I'm pleased to say that just in a few short years, Lee Rubin's made enormous progress for this on spinal muscular atrophy. We're now collaborating with various pharmaceutical companies to try to get those chemical leads into the clinic. And we're so excited about this program that we're now expanding it very aggressively into numerous diseases, into Parkinson's, into Alzheimer's, into diabetes, to spinal muscular atrophy, I already mentioned, into muscular dystrophy, because we're convinced that this is the best way and a new way to discover drugs. The reason it's the best way, I'll remind you, is it's not looking for drugs in rodents, it's looking for drugs on human cells. Secondly, it's not looking for drugs by using the cells that you can easily get from a patient. It's looking for drugs that affect the cells that are defective in the patient. In the past, people only screened for drugs on skin cells, even if they knew the disease affected nerves. But now we have a way to make the cell that is defective in the disease. Okay, if I haven't depressed you about all the diseases you're going to get and how long it's going to take to cure them, let's set that aside and think about something happier, about healthy aging. So I just draw a line here to say that scientists are very excited and very active now on using stem cells for cell replacement and as tools to get at the root cause of these degenerative diseases. And I'll be glad to answer questions about that at the end. But I now want to turn to a different problem. It's not such a problem, you could say, but it's a fact of life. I often note this story that I don't like doctors, I don't like going to doctors, I should say. I don't imagine anybody does. But when I turned 50, I was told I had to start going to the doctor, and I hadn't been to one in a long time. And the doctor said something that kind of annoyed me. He said, "You're in extremely good shape for a man of your age." And it was this man of your age thing that sort of troubled me. So I saw this cartoon, which I quite like, as we think about healthy aging. I'll read it to you for those in the back. It says, so in general, I'm a very optimistic person. I don't like the idea that I'm now going to just think about what disease I'm going to get and learn to live with it. So let's think about what I've already told you about animal development. We come from a fertilized egg, stem cells renew and replace our body. Why can't we harness that natural ability of stem cells to regenerate and replace our bodies and keep them healthy? This is a really simple idea. Our body does it, it just does it less and less effectively as we get older. So let's have a look with some cartoons at the characteristics of aging. So here's a young woman, an undergraduate age, you could say. And then as time goes on, she will slowly age, an attractive older woman. What are the properties of that? Maybe I'll show you that again. What are the properties that correspond to aging? Well, if you think about it, they're sort of obvious things because many of you have already started to experience them. As a young person, your organs are very robust, wounds heal rapidly, and there's high tissue turnover. As we age, that slowly degrades. Our organs become more frail, wounds heal slowly, there's low tissue turnover. I think it's worth thinking about stem cell science, not to prevent that from happening in the extreme, that is not to make everyone immortal, but just to slow the process and make you enjoy your later years more actively. So how might we do that? Well, here I'm glad to tell you about one of my favorite experiments from the Institute done by a young woman, Amy Wagers. She would not describe this experiment the way I do, but I call it looking for the fountain of youth. And what Amy did is have a very simple idea that we were able to support with our seed grant program, where she said, well, if young and old animals have this difference, how would you go about finding the stuff responsible for that difference? And the way she did it was quite simple. All good experiments often are very simple in their design. She took a young and an old animal and put them together so that they shared their bloodstream. The question then being, is there something in old blood which is like a poison, which makes you repair your organs less well? Or is there something in young blood that does it that just is missing from old blood? Now, the way the experiment is done is one cuts a skin flap, say like through my shirt here, and then I would sew myself to another person, and the two people then share a bloodstream. The blood flows quickly, courses through both animals. So they now have a common bloodstream, mixed of young and old blood. And the mice, I can tell you, don't mind this at all. They still run around the cage quite happily. So the experiment she did then, so you could ask, well, what could you test with this? You might want to test really interesting things like memory or perspective on life, but of course mice are too stupid to ask them questions about that. So we're testing, to start with, things that are much more manageable. And the example I'm going to show you is about muscle repair. And to introduce it, I'll remind you that when you were young, if you bumped your knee or fell off your bike, you probably didn't even remember it a day or two later. If you go out for a nice bike ride this afternoon and fall off your bike, you'll probably remember it for a couple of weeks, because it takes your body a while to repair muscle. I'm going to show you pictures of how muscle repair looks. So here we're looking for signs of regeneration in muscle of old and young mice. So let's begin here, and this is really like the biology lectures. I'm going to show you the control experiment and the result. So if we take two young mice, so Y here stands for young, and injure the thigh muscle in one mouse, it repairs its muscle very well, and that's seen by the amount of red cells here. So where there's red, that means repair. This is a section through normal muscle. We're looking down the end, like down the end of hairs. These are muscle fibers, and the red means the muscle has renewed itself and been repaired. If you now put two mice together where one is old and one is young, and you now injure the young muscle, it repairs at the young rate. This is an important control because it says that old animals do not have a poison in them that blocks repair. If you put two old animals together, you see very little red. They're incapable of repairing their muscle at a fast rate. But the wonderful result here is if you put a young and an old animal together and now injure the muscle in an old mouse, it repairs at the young rate. This is a fantastic result. This means there's something in a young animal that's missing in an old animal that we should be able to isolate that will cause faster muscle repair. Amy has gone on to show that that faster muscle repair does not depend on cells from the young animal moving to the old animal, but instead it's some substituent of the blood. So now it's pretty obvious that what we want to do is develop a simpler, faster assay, an in vitro test tube assay, to find out what's in the blood of a young animal that's making muscle repair. So these are the three things then that I wanted to tell you about on the science side, about why scientists are excited about stem cells. I often make the analogy that it's hard to predict where this science will go, but the analogy, I would say, is a reasonable one with the discovery of the transistor. Before the transistor, you'll remember all electronic devices depended on a vacuum tube. And when the transistor was discovered, no one predicted that half of you in the audience would be glancing down at your BlackBerry or wondering if you've got a cell phone call, or knowing when you get back to your room, you'll go to your personal computer. All of those things came from the transistor. I can't predict with certainty what will come out of stem cell science, but it is going to change biology and medicine, essentially because we now have the unit of life, the cell, in a test tube, and we can manipulate it. Now, with that, you could say, well, my gosh, why isn't everyone working on stem cells? And so now we come back to the complicated issues, which I have skirted and indeed skipped over. And they have to do with the question of where do these embryonic stem cells come from? So I'm going to finish up talking about this and then open up for questions. Stem cell research raises the very reasonable question, an important question, a question that I think is important for public debate. Is embryonic stem cell research morally objectionable? This sort of begs the question of what is the moral status of the early embryo, the embryo I have shown you there in a picture. So let me tell you now where embryonic stem cells come from. This is what it looks like in a cartoon, and I'll show you a real picture in a moment. but I have to remind you about early development. So a fertilized egg will divide in the first few days into a ball of cells, which has the technical word actually is a morula, which comes from the Latin for raspberry, because it kind of looks like a raspberry. And then it develops a little farther, kind of like a tennis ball, where there are cells on the outside, and then there's a cavity on the inside. And the cells on the outside will make the placenta, and the cells on the inside make the human, the embryo. This is true in a mouse and in a human. In order to make an embryonic stem cell line, what we call derive an embryonic stem cell line, we have to isolate the inner cell mass, which destroys this. It eliminates its potential to ever make a person. We then culture it in a petri dish and grow out embryonic stem cell lines. In real life, that looks like this. Here's a frozen human blastocyst, and here are colonies of embryonic stem cell lines. So blastocyst to human embryonic stem cell colonies. Over the years, the Harvard Stem Cell Institute has been the most prolific at doing this. We've isolated more cell lines than any place in the world and send them out to researchers all over the world for free. And we're now able to do that with about a 50% efficiency. So far, we've made 76 of these lines in collaboration with several in vitro fertilization clinics. So let me now talk about where we get the frozen human blastocyst. And I want to say this clearly because I still get a fair amount of hate mail about killing babies to make embryonic stem cells. Couples, as you may know, often want to have a baby, and if they can't do that by what I would call natural means, they sometimes seek medical assistance at an in vitro fertilization clinic. The couple has to donate the two key cells to make a fertilized egg, sperm and an unfertilized egg. The unfertilized eggs are usually donated always following hormonal injection, but usually in a number of about 10 at a time. Those 10 eggs are then fertilized, and typically 2 to 4 of them don't survive very long. They die and are discarded. Of the remaining ones, some are implanted into a woman's uterus to make a baby. At some point, that infertility treatment is concluded. Either there's a baby or there isn't, and there are almost always leftover fertilized eggs. It's estimated that now in the United States there are about 400,000 fertilized eggs in freezers, and one of the most active areas for that research is here in Massachusetts, and so the IVF clinics with which we work are local. They're all within the 128 area. And they have literally thousands, tens of thousands of these frozen fertilized eggs. In 2 rounds of informed consent, the couple is then asked what they would like to have done with the frozen fertilized egg, which unquestionably has the potential to become a person. They can either keep it frozen forever, they can discard it if they're tired of paying the yearly fee to keep it frozen, or they can donate it to another couple to make a baby, or they can donate it for research. If they say they're interested in donating it for research, they then ask to think about it for another month, and then asked again. And it's after that process that our laboratory receives those and derives human embryonic stem cells. My own view of this is that this is perfectly ethical research. It respects the quality and dignity of life, and it's trying to use science to help people that are suffering from disease. There are people who disagree with that view, and that's led to what I would call, maybe my most complicated slide here, the back and forth on stem cell science. The bottom line here, I'm going to go through this slide in a minute, but the bottom line is that we're living in a country that is unable to make a clear decision on whether this work should be done with federal funds. So the background begins in 1995 with an important piece of legislation called the Dickey-Wicker Amendment. This bans the use of federal funds for any experiment in which a human embryo is either created or destroyed. Now, it's important to note that that law, that amendment was passed 3 years before the discovery of human embryonic stem cells. And the amendment is ambiguous, in my view, on whether or not one could use federal funds for this research. The federal funds aren't used to create the embryo, and they need not be used to destroy it. But we would dearly like to use federal funds to work on the cells derived from that. President Clinton decided that no federal funds could be used, therefore, to derive the lines, which does involve the destruction of the embryo, but could use federal funds to study them. President Bush said that was too permissive, and one should only use those lines that were in existence before he thought about it in 2001, and then made 5 lines essentially eligible for federal funding. President Obama, more than a year ago, said that was an unnecessary restriction. If the work is ethically and morally responsible, why not do it full out rather than with just a handful of lines? But you may have seen that in the last few months there's been a federal court case which is now calling this all into question. And I'm told that it's absolutely unclear how this will come out. My own view is that a legislative solution is required to clarify what could and should be done. And it's also my view that that's unlikely to happen in the present day construction of our legislative bodies. I'm going to finish and answer questions with a couple of final points that I'd like to make. First is, all of the work I told you about today, I've only had some small part in it. In my own lab, I thought you might be interested to see what a lab like mine is composed of. In my case, it's composed of about 20 to 30 people. These are medical doctors, MD, PhDs, and PhDs shown here on the left. And then there are 2 graduate students. I take rather few graduate students because in my view they require and deserve a lot of my attention, and so I prefer to have mostly postdocs and a few graduate students. But I love having undergraduates in the lab, and these are all the undergraduates who are working in the lab now. But my lab is one example of a large institute here called the Harvard Stem Cell Institute. And that is an institute that composes about 630 researchers in the Boston area. It's people at the medical school and the college where I work, and all of the affiliated hospitals as you see here. So one thing I think is just wonderful about Harvard and these hospitals is they've joined together to share information, to cooperate, to encourage young people, to provide seed funding, to make this science move forward. In fact, this institute would not be nearly so successful, you may be surprised to hear, if it was not for the Harvard Business School. And that's in part because of at least 2 people here, Bill Solomon and Howard Stevenson, have been enormously helpful in setting up the structure of the institute and in bringing to us generous philanthropists who are business school graduates who far and away provide most of the philanthropic money that we are able to put, I think, to good use at the institute. So when they asked me to come to speak today, I was delighted to do it because I'd like to do it in part by way of thanking the business school. The institute is really quite unusual. Things like this didn't exist before, and there are 2 examples of it here at Harvard now. One is the Harvard Stem Cell Institute, as I've been preaching to you today, focused on the use of stem cells to find new medicines. And there's another one in the Harvard now, the Hans-Jörg Wyss Institute, the Wyss Institute, for biologically inspired engineering. And both of these, I think, show a new way for Harvard to act as one unit across all of its schools and take on important problems that are not just theoretically interesting, but have a real impact in the world in the longer term. So I've saved plenty of time for questions. I'll just end by thanking you for coming, and I'm glad to take any questions today. It's a little hard for me to see hands because the light is in my eye. So there may be microphones down here, or if you have a very booming voice, you could just shout out.
I have a very booming voice. Just a quick technical question. Pluripotent or multipotent stromal cells, are they anywhere, it's not embryonic, right? Those aren't embryonic stem cells. And so do they bypass these rules, and are they robust enough to be used in this research?
The question, thank you for the question. The question was about other kinds of pluripotent stem cells. The only stem cells that can make any part of the body are embryonic stem cells. And these pluripotent iPS cells I described are likely to be able to do that. The other kinds of stem cells you hear about, like cord blood stem cells or amniotic stem cells or mesenchymal stem cells or stromal stem cells, those are all stem cells, but they have a limited potential. So they can, in general, make things like connective tissue and fat and muscle, but they can't make eyes or pancreatic beta cells. Now, when I say can't make it, that is with our present understanding. But it's sort of obvious to you that I would not have predicted 10 years ago that we could take a skin cell and turn it into a cell that has kind of reveal its latent potential. But thank you for the question. It sort of I might extend it a bit to remind you that many people call me and say, well, not many, some people call me and say, we're about to have a baby, should we save the cord blood? Because it says in the brochures that cord blood stem cells are pluripotent and can make any part of the body. What the brochure actually says is technically true, but it's wrong. The brochure says some people say that cord blood cells can make any part of the body. Well, some people do say that, but they're wrong because it's never really been demonstrated. And so I don't mean that one shouldn't freeze cord blood cells, because it could be that in some years we'll discover ways to make better use of them. But at the moment, if I had to predict what is the most useful cell, it is the human embryonic stem cell, because it's natural and normal. But this iPSL is coming up quickly, and I wouldn't be surprised if in a few years we can use it for almost all of our experiments. Maybe not for transplantation, but certainly for all the drug screening. Sorry, I think we'll have to use the microphone because we have two people talking at once. Well, three, counting me. Sorry, could you repeat the question? What about? Yes, the question was about the role of stem cells in cancer and could it be used as a cure for cancer. It's a very good point, which I didn't bring up today. So if you'll bear with me for a little bit of biology on cancer tumors, when a tumor starts growing uncontrollably in the body, it almost always has two kinds of cells in it. These specialized differentiated cells that I talked about, all the cells of our body. But usually it doesn't make all of them, it just makes a few of them. So if you have a muscle tumor, it might be making more and more muscle. There's a very peculiar kind of gory kind of tumor called a teratocarcinoma that actually makes grapefruit-sized tumors that have teeth and hair and parts of eyes. So it shows that cancer cells have all that potential to make everything. Those, let's call those the specialized cells, and those are the cells which are the target of chemotherapy and most radiation therapy. But there is unfortunately another cell within many tumors, maybe within all, called a stem cell, a cancer stem cell. And that's like the seed that keeps making the tumor grow. So to a first approximation, one explanation for remission in cancers is that the treatment has killed off most of the specialized cells, but not the stem cell. And then over a period of time, that seed grows more and more tumor. So your question is a very good one. Can we use that information now, combine it with what we're learning about stem cells to target specifically cancer stem cells? That's a very good idea. It's not in the clinic at the moment, as far as I know, but I wouldn't be surprised if we see more and more specific attacks on stem cell specific parts of cancers, for example, like rhabdosarcomas, to find the cancer stem cell there and then target that with a specialized kind of therapy. Yes?
Assuming the federal court rules against the use of embryonic stem cells, how will that affect Harvard Stem Cell Institute, number one? And number two, how much has, or have the political and legislative problems that have existed in this area over the last 10 years retarded the advancement of stem cell research in the United States vis-a-vis other countries around the world?
Okay, the first question, thank you for those questions. The first question was, what is going to be the effect if the Supreme Court, where I imagine this case will eventually end up, decides that the Dickey-Wicker Amendment is so ambiguously worded that it is unreasonable to use federal funds? That would be a perfectly reasonable, I hope that's not the outcome, but it wouldn't be an illogical outcome of that case. The effect of that means that the work is not illegal, and I probably should have emphasized that. It's that you cannot use federal funds to do the work. And that relates to your second point. So we here at the Harvard Stem Cell Institute, with all of the affiliated hospitals, banded together to work on this, and the effect of the last 10 years has simply been to slow down the work. It hasn't been to stop it, it just slows it down. And so what you'd like me to say, or what you'd like a way of answer is, well, by what factor? Is it gone half as well? Has it been slowed down by a factor of 10? I can't really give a reasonable number to it because this kind of activity depends on discovery. And discovery is sort of like, popcorn. You don't know where it's going to come from. But I could say it this way. If the ideas that we have that our executive committee reviews for our funding are good ideas, and I think the evidence for that is in and is quite convincing, we could easily do with 3 to 5 times as the resources we now have. Not 10 times, but 3 to 5 times. So I would guess that if we hadn't been playing this Mother May I for the last decade, I would be telling you about at least twice as many discoveries today. Does that answer the question? Oh, no, I didn't answer the part about foreign countries. So that's not really an easy comparison either, because the US is so vastly far ahead in terms of research spending on biomedicine that we had a sort of big head start on all other nations. Nevertheless, there's very significant work going on in several nations, in particular the United Kingdom, which has very clear and sensible laws on this matter, in China, in Japan to some extent, in Australia, in Singapore, in Israel. I might just note, to go back to the dark side of it, there are other nations that have decided this work should not be done. In Germany, the discussion quickly turned on the relationship between this and the eugenics movement, on controlling the genes of people, and it's illegal to do this work in Germany. In Italy, the work turned immediately into a discussion of the role of the Catholic Church, which is opposed to all IVF treatment and has made working on stem cells a new kind of, I think it's called a venal sin. Actually, my mother would be angry that I don't remember that, because I was raised as a Catholic. I can't remember what kind of sin it is, but it's a sin to do stem cell research. And I think you can see the puzzle. How would I describe it in this country? I would say that if I'm going to, in a superficial way, connect stem cell research to eugenics in Germany, in a superficial way, I would connect it to abortion in the United States. It's connected to the tortuous politics of abortion, which we've all lived through for decades, and I don't see an easy solution to it. Again, I'm sorry, I can't see because of the light in my eye, so.
I have a question also comparing the work that's being done in the US versus, for example, China. I live in Hong Kong, and I've had a number of friends who've had all different types of diseases, went to the best specialists, I gather, in the world, and ended up in Beijing in a stem cell or medical hospital or institute. One of my friends went to visit them, and their progress was quite remarkable. I hesitate to say that they're cured because I really know nothing about the field. But in terms of being able to continue having, how to say, a quality of life, which they were not able to have before they had this treatment in Beijing, it was truly remarkable. So I was wondering, are you familiar with the work that's going on in China and Beijing? Is it, slanted in a certain way? Is it robust? I know they've also actively courted a lot of the postdocs and the researchers here at Harvard. They flew some young man all the way to Beijing, gave him royal treatment, and in the end, the young man did go and work. The other ones decided to go to other institutes. But I'd be very interested in your perspective in that.
Yes, well, there are a couple of points to your comment and question. I'll begin by saying that we have extensive interactions with China. You wouldn't have been able to tell, but just as one example, about a third of the postdocs in my lab are from mainland China. And the Harvard Stem Cell Institute has regular exchanges with our Chinese colleagues. One of the things I said but didn't emphasize is our view about this science is very much a kind of, well, in the computer industry you call it an open source model, that all of the things we discover and create by way of reagents, we make a special effort to give out quickly and freely to all researchers without restriction the world over. But you asked about particular treatments. I can say that because of the excitement of this science and its visibility, I should say, I'm sorry to say that in many places there are doctors doing treatments for which there is no scientific evidence. I don't know in a public forum what's the polite way to call that kind of snake oil treatment, but I'd just say that, for example, taking mesenchymal stem cells for cardiovascular disease doesn't have a sound scientific basis. At the same time, one should remember that sometimes you try things and you don't know why they should or shouldn't work, but they do work. And then there's a long period of discovery to figure out why they had a positive effect. You mentioned some friends of yours which appear to have had a positive effect from some stem cell treatment, and I'm delighted to hear that. But before one could draw a conclusion, one would of course have to have more patients and do controls, right? So to have them not take the treatment, people who had a similar condition. It's early days in this science, and I would say the only stem cells for which there's incontrovertible evidence of a positive clinical effect are the transplantation of bone marrow blood stem cells and the transplantation of skin cells, which I didn't talk about today, for burn victims. That doesn't mean that other things aren't working. I see when I'm at the grocery store that Liz Taylor gets goat stem cells and it makes her feel better than ever and things like that. I have no real way of commenting on that, but if you ask my advice, I would not go for the goat stem cells. I just wouldn't do it. Yeah. I see a question here. It's a great question. It's good I speculate, which I like to do. So this is, you can't hold me to this. But I mentioned the Wyss Institute at the end, which is focused on bioengineering, and you brought up the idea of nanotechnology. My long-term vision would be that we will learn enough about these cells to make not just cell types, but tissues and then organs. And how are you going to get those into people? You need very sophisticated kind of bioengineering, which we do not now have. So we have, as you probably know, various kinds of prosthetics for bone and some joints, but we don't really have good ways of making muscle. So let's say you wanted to make, my little right bicep. How would you do that? That's what I would like to speculate about, is how do you make body parts? In fact, you might even think that some of the movies you've watched, like Terminator or something, aren't so fantastic after all. Will there come a time when we can replace parts? Then the next frontier, of course, would be our brain. So even if we knew how to make all the brain cells, we have no clue about how they're wired. And I like to think about the puzzle about downloading information. I was thinking about this the other day. You'd have no reason to know that my wife and I became the masters of one of the Harvard undergraduate houses called Eliot House. And I was told I had to learn the names of 430 undergraduates. This is not an easy thing to do, I've discovered, okay? And so if I had if I could take my little, Macintosh dongle and put it in here and download their picture and their name, that'd be a big help. I would go for that. Yeah.
In the middle, Doug.
If you readily share the science with the world, how does Harvard protect their intellectual rights for patents and future business applications?
Good point. So if everything we discover, we say, everyone come and use it for whatever you want, how do we gain any benefit from the investments we've made? I think, for me, I have to tell you that the gain is helping people in the end. But let's be realistic about that. You can't do that without protecting the intellectual property. So we're very aggressive about patenting things. For example, the new method I just described to you that Derek Rossi developed about how to make iPS cells from skin cells without genetic modification has been patented and will likely be licensed to some pharmaceutical company or be used to start a biotech company. There have already been several spin-out biotech companies from the Stem Cell Institute. I'm less familiar with what's come out of the Wyss Institute, but I know they share the same view. The other side of that, sort of the scientist and scholar in me, likes to dream about a different thing, is that one of these patents will be so valuable, I will never again have to write a grant. That would be like the dream come true. So I like to talk about this with Howard Stevenson, who many of you know. We came up, I think, with the term of a Medici fund, because you'll remember the Medicis said to Galileo, you're doing interesting stuff, just send us the bill. Like if you want a telescope made or just do that. For a scientist, that would be the dream. You may not appreciate that scientists spend an enormous amount of their time writing grants. It's worth sort of contrasting that when a hospital hires a doctor, they don't tell the doctor, well, now you should spend 30 to 40% of your time writing grants so that you could treat patients. In effect, that's how universities work. They hire scientists, but we spend 30 to 40% of our time literally writing grants and filling out reports for them. That's not going to change quickly, but it is one of my dreams that something will be discovered from the Institute. So there would be an evergreen fund so that young people can come when they have an idea like Amy Wagers had or Derek Rossi and just do it. In fact, I've heard that our new dean here at the business school, Dean Noria, has this interesting idea of an innovation center. You may have heard more about it than I have, but it seems to me like just the kind of place where you'd like to take the world's best undergraduates and graduate students and business students and say, think big. How can you change the world?
Dr.
Melton, can you comment on the application of stem cell research to inflammatory bowel disease?
Yes, the question is about stem cell research for inflammatory bowel disease. There's no immediate clinical work being done now, but the reason that for your question, which is a good one, requires me to tell the rest of the audience a little bit more about the intestine. So the intestine is a very unusual organ in that it turns over extremely quickly. I should know the rates in people, and I don't, but I know in mice, the cells inside the intestine are little finger-like villi that absorb the food, turn over in less than 2 weeks. It's a very rapid turnover. And the cells are replaced by an intestinal stem cell. And so in inflammatory bowel disease, there is, in some cases, an immune attack or some problem with something called autophagy in those cells that leads to these problems. And one likes the idea at the moment of purifying the intestinal stem cell and then asking, can we sort of boost its activity? Can we make it work faster and better? And we don't have a program in that at the Stem Cell Institute now, but it's an area where I think there's significant opportunity. In fact, your question reminds me that I didn't explain how do we pick which diseases we work on. It's a sort of an interesting ethical puzzle. You could say, well, you want to pick the diseases where when you get it, there's no treatment and you're going to die. Or you could pick diseases that affect children. Or you could pick diseases that affect your family. All of these are legitimate criteria. In the end, we pick diseases where our executive committee sees a very clear, sensible, systematic path to the clinic, and we have good scientists that can work on it. That doesn't mean that there aren't diseases to which stem cells could be applied. It just has to do with the limitations of our resources and what we have here now. And I should point out that the Harvard Stem Cell Institute is far and away the largest concentration of stem cell scientists in the world. It was recently judged, I forgot what journal did this, an independent journal, looked at 16 aspects of regenerative medicine and stem cell biology, and the Harvard Stem Cell Institute ranked #1 in 13 out of the 16. So at this time, it won't always be true, but right now, if you're a young person and you want to work on IBD, and it doesn't really matter where you are in the world, you try to get to the Harvard Stem Cell Institute. It's sort of where the action is. In fact, if you're ever around, you'd be delighted to see our, bi-weekly meetings which attract up to 200 students. It's just so exciting to see the people thinking of all the things that could be done.
If I understood correctly, you said that within 5 years you'd be able to produce an islet or beta cell, a human beta cell. That means producing the cell, and after you go through the clinical studies, what would you estimate the time to actual clinical use?
You would think I would be better at answering this kind of question because I get it all the time, which is, how long will it be before all of this gibberish you're talking about will make a difference to me? All I can tell you is this, maybe if you'll allow a personal intervention. My 2 children have type 1 diabetes, and they ask me that all the time. So I come home and I say, Sam, I'm so excited. We've solved one more step in this process. And Sam really wants me to tell him when we're at the end. And so I don't know, I can't tell you with any certainty when that will be, but I would be deeply disappointed if it wasn't in my lifetime. I think it will happen. I repeat what I said earlier. Everything we're learning about this tells us it's a solvable problem. And I would not say that about every disease. I'll contrast it with the most unfortunate disease of autism. I'm not sure that's a solvable problem. It could be the way the brain is wired. Once it's miswired, it's very hard to repair. So I'm not here telling you that stem cells are going to cure every disease. But the diseases I talked about today are solvable problems. So I'm sorry, I'm avoiding your question. I can't answer when it's going to happen.
Doug,
Howard.
you might inform the audience sort of what your budget is, because I know the VEAS is trying to spend about $15 million a year on happy or experimental things that the NIH won't fund. You get a lot of funds from NIH and Howard Hughes, but
Right.
they really, truly, things that are pushing the limit are not easily funded. How much are you spending?
That's right. So Howard's asking for estimate of the budget, as a good business school professor would. And so the total cost of the research, I don't know the answer, because you'd have to include the cost of the buildings, all of the infrastructure, some of which is supported by the NIH and the university, the Howard Hughes Medical Institute, and others. But the projects that make a real difference, the things that come from our junior faculty program, from the seed grants, the things I talked to you about today, we spend this year a little over $18 million. And all of that money comes from generous philanthropists.
Here. Hi, Professor. My question is also regarding somewhat diabetes. So you mentioned that type 1 diabetes, it's a stem cell would help produce beta cells that would then generate insulin. What about diseases such as type 2 diabetes, where cells have become somewhat resistant or no longer respond to insulin? How could a research such as stem cell help develop these cells or aid in this research?
So the cause of type 1 diabetes is at some level better understood. There's an autoimmune attack. The body's own immune system kills its own beta cells, so the patient is left without any beta cells and can't make insulin. The other type of diabetes, sometimes called adult onset or type 2 diabetes, has a less clear cause. People in the United States in general believe it's a disease of insulin resistance. The body's making insulin but not responding to it. In Europe, it's generally believed to be a disease of insulin insufficiency, the failure to make enough insulin. And you can imagine that if scientists can't agree at that sort of basic level, it means we don't really understand the cause. What everyone agrees on is that there's a significant period of time, maybe a decade or more, during which the body's capacity to make insulin is being strained. The evidence suggests it's being strained by two things: lack of exercise and an unhealthy diet. During that strain period, the beta cells essentially, and I'll use a very technical term now, poop out, okay? They can't make enough insulin to meet the metabolic demand. So stem cells are relevant to type 2 diabetes in two ways. One is if we could figure out a way to stimulate the replication of pre-existing beta cells, that would mean in the sort of decade or more before the person requires oral antidiabetics or while they're requiring them, and certainly before they would require insulin injections, you could boost their insulin production. The second way would be the way I talked about today, which is to take a type 2 diabetic, isolate a skin cell, make a stem cell, and then make their very own beta cells for transplantation. That, of course, has the enormous advantage that endogenous or natural beta cells will control blood sugars much better than any drug, any insulin injection will ever do. That means that the population there, to get back to numbers we're talking about, is something like 40 million people.
Would you say, you mentioned muscular dystrophy, would you say that there is a chance for young people that are 13 to 15 years old that have the muscular dystrophy, the Duchamps, do something?
Duchamps,
Yes. Do you think that there would be possible that there would be some hope for a cure for them?
I would say it more strongly like that. I think there is great hope for a cure. The work I didn't tell you about today involves the isolation of a cell in the muscle called a satellite stem cell that makes skeletal muscle. And again, the same young researcher, a spectacular person, Amy Wagers, has identified that cell, purified it, and shown that if she injects it into a mouse model of muscular dystrophy, she can repopulate the muscle and make it 90% as good as wild type with these satellite cells. She calls them SMPs, skeletal muscle precursors. So there's great progress in that field. But of course, we're accustomed in our modern society to think of things on a different pace than the way scientists think. So let me remind you how scientists think when I say there's great promise. This isn't like, how would I say, I'm always amused when a reporter calls me and says, what's new? And I say, well, I just talked to you a month ago. I mean, for them, they're looking for something new every day. If we have a good idea once a year, that's a pretty good rate of having a good idea, and it takes that long to work on it. So when I say there's great promise, I don't want you to think I mean next year. I think there's a path forward, there's a clear plan for how you could think about treating the disease, but it's not yet ready for clinical trials. It's in the stage of treating mice with this new approach.
If you had a 15-year-old son that had this, where would you send him?
Well, I'm not a medical doctor and I don't work on that disease, so I better not
What research? I mean,
if you wanted to talk to someone about the exciting research,
yes.
I would talk to Amy Wagers, and I'd be glad to give you her contact.
Okay, thank you very much.
One more question. I'm sorry, I can't hear you. So could you get the microphone?
Hi. At what capacity do you think you're running your lab? In other words, if you had all the funds in the world, how much more research could you actually be doing now?
I'm very confident about saying that the Institute would not have any decrement in the quality of the work it's producing if we tripled our size. That is, tripled our budget for these sort of seed things. But we couldn't make good use of 10x. We had hoped that there would be a big building here across the street in Alston, which would have allowed me to say maybe 5-fold. But given our space now, I think we could easily and very effectively use 3 times the resources we have. And in general, I'm conservative about those things. So I thank you all again for sitting in this dark room, and enjoy your weekend.